Hi, I'm Rachel Owen, co-founder and director of Most Policy Initiative. Our mission is to give policymakers better tools for implementing policies backed by science. We want to be a trusted resource for scientific information and expertise. This is our first virtual webinar about the challenges facing our state. We partnered with Missouri Cures Education Foundation to go over a brief history of COVID-19 and explain what scientists around Missouri are doing to help. First, we talked to Dr. Daniel Hoft at St. Louis University. He was one of the first called into action to fight the pandemic. He'll explain how he found out about the SARS-CoV-2 virus and how he's helping to create a vaccine. Next, Dr. Michelle Altrich in Lee's Summit explains how ViralCore Eurofins is leading the way in testing for COVID-19. At Washington University in St. Louis, Dr. Philip Payne tells us how new approaches to data and crunching the numbers are helping Missouri's decision makers reopen our economy. Finally, researchers at Missouri University of Science and Technology are working on a project that can make air transportation safer and healthier for everyone. Without further ado, let's get on with the show. Here's Dina Ladd, Executive Director of Missouri Cures, interviewing Dr. Daniel Hoft.
So with us is Dr. Dan Hoft. He is a professor in the Department of Internal Medicine, and he's the director for the Division of Infectious Diseases, Allergy, and Immunology at St. Louis University. So Dr. Hoft, thank you so much for joining us. Can you walk us through when you were SLU and you were approached and told, oh my gosh, we have this coronavirus and we need you to play an important role in this and what you're doing and so forth?
I heard that there were some people dying from potentially a new virus in China at the very beginning of January, but we didn't know the significance of it at that point. Mid-January, I went to Santa Fe to go to a tuberculosis research keystone meeting. And there were about, I think, 20 participants, something like that came from Wuhan. So we heard a little bit about it there. Nobody was sick, so we don't know if anyone got infected at that conference, but I became a little bit more aware. I went straight from Santa Fe to DC for the kickoff meeting for our new VTEU network. And Dr. Fauci loves to give a speech for this network of PIs that get together for this annual meeting. The night before, he had been called by people that didn't ask, but told him he had to be at meetings at the White House and in Congress because of this outbreak. And that's when it really hit home that there is something significant going on. He said, I'm sorry, but I have to leave right after I finish my talk because we have to go deal with this, and you are going to be tried by fire. Within a week or two, we were being asked if we were interested in participating in a treatment trial, first comparing placebo versus remdesivir. That was just announced. The great news that there was evidence that remdesivir, that's from this trial, was superior to placebo. So what we found was that the number of days that you were sick after starting remdesivir were about 2/3, a little bit longer than that, like from 15 down to 11 days was the median or average time for how long people were sick. 15 days placebo, 10 or 11 in remdesivir group. In addition, there was not a significant, but a trend for reduced mortality in the remdesivir group. There's many vaccine candidates that are in the pipeline. One of the leading ones in the beginning is this vaccine produced by a company called Moderna, which collaborates with the Intramural Vaccine Research Center at the NIH. And they have developed a novel type of vaccine called an RNA vaccine What that means is you're not actually injecting a purified protein. You're taking a step back. Proteins are made by having, you have to have DNA, that's your genes, that encodes everything. And then you have to make RNA copies from the DNA. The last 5-10 years, people have realized that we can use the RNA as a vaccine without having to actually make the protein and purify it and make sure it's stable and all that. People have been working for 10-15 years on this and had applied this RNA technology to MERS, a vaccine that they were developing, and had learned that for coronaviruses, the spike proteins were important because they were involved in binding to cells to initiate infection. But that it was hard to get a good immune response that was protective against them because the spike protein normally has a different shape than when it's coming in contact or about to come in contact with the cell it wants to infect. It actually pulls back its sheath from this protein that is covering the site that actually binds to a cell that's being infected, becomes infected. So by exposing that, by what they did was they changed the sequence of the RNA just in a few different places so that those sheaths were pulled back all the time, and that binding site that binds to the cells that the virus is trying to infect can engage. But as a vaccine, when you produce this protein from the RNA, the binding site is exposed to the immune system all the time, so it induces an immune response against that part that's normally not seen by the immune system, and only temporarily during the actual infection event. And within 3 to 4 weeks, they'd already made a GMP lot of vaccine for SARS-CoV-2. Just amazing timeline. And within another month, the first vaccination was given to a human volunteer. Because of the EUA, the emergency use authorization option from the FDA that allows rapid licensure in the face of a deadly threat, like a pandemic, this vaccine was able to go kind of from in silico, the computer design and synthesis of the sequence, and then putting it into the viral vector that carries it, and the lipid nanoparticle formulation that encapsulates it. That could be done rapidly, and that could then get into people. And phase 1 is not quite over, but it'll be going on for a while. But the key safety data is almost all collected, so that we'll be able to, before the phase 1's over, be able to move forward with phase 2 in this EUA. Wow. We're expecting that to happen in a couple of weeks to a month.
Today we have Michelle Aldridge she is the president of ViralCore Eurofins. So thank you for being a part of our Missouri Fighting COVID-19 video.
Absolutely, it's great to be here today.
So can you tell us a little bit about ViralCore Eurofins and why you were positioned well to respond to the COVID-19 needs?
Absolutely. So here at ViralCore, we have a long history of serving critically ill patients from an infectious disease perspective. And actually, we've got a very experienced team of R&D scientists that have responded to infectious disease crises before. Most notably back in 2016, when we had the Zika emergency be declared. Of course, a very different pandemic than this one, but very severe in its own way. And then back as early as 2009 with H1N1, we also developed and were one of the companies that worked with the FDA very early on to get a test to market to diagnose that disease. And so we recognized here at ViralCore the need for the test. way back in December, when we had the first reports of this novel infection coming out of China. And so early in January, we reached out to some very distinguished infectious disease physicians that are part of our medical advisory board and talked to them about what they knew about this virus from China. And they said there isn't a diagnostic test on the market that will pick it up. So right there, we knew that we would have to have new techniques to detect and diagnose this disease. And so we really got to work, and we started to leverage really the entire Eurofins network. And so here at ViralCore, we're one clinical lab owned by Eurofins, but we've got other clinical labs throughout the world. And so we started working on collectively as a group in building different assays to make sure that we would have one that would be ready for market when the need arose. We've got a really dedicated team of laboratory associates and support staff here. You know, they're really proud to be able to support a solution because it really has, one of the great things about the pandemic is it has allowed us to interact with a lot of organizations we hadn't before. And the teamwork and the partnership that we've seen has been amazing and has been great. And it is something that we can leverage in the future once we kind of get through this, and we will get through this. It's going to be a long road, but life will return to normal at some point. And so it, you know, just understanding kind of all these new connections that we've been able to make, and how much stronger we can make our public health system after this to be able to respond to future ones in a much faster mechanism. And really, when we look at the history of that pandemic, things really changed on February 29th. And so before that, since an emergency had been declared, the FDA mandated that they review and approve all testing before it can be offered for patient testing. But on February 29th, in response to a lot of feedback from clinicians, they modified those rules, and we were then allowed to offer the testing once we had validated it to CLIA and CAP standards, and then work with them post-launch to get that emergency use approval. And so we did that very quickly. We started testing here at ViralCore on March 13th and had full EUA approval by April 6th. And so really a very quick time to bring a test to market.
So your company has over 30 years of experience working on infectious diseases. How does your response to SARS-CoV-2 compare to other diseases you've handled?
Sure. So when we think most recently about Zika, you know, I think the difference with Zika really is in the scope of it and the types of patients that it was affecting. And so it really, in some ways, was very similar. And so kind of our ability to respond to Zika, really, we were able to leverage that when we looked at responding to SARS-CoV-2, because both are RNA viruses, and we were able to leverage a lot of the techniques that we had developed to measure and diagnose Zika with SARS-CoV-2. Now, it did have a lot of differences, of course, because Zika is transmitted by mosquitoes, and so the range of people affected was just very different. And of course, most notably now, the scope of the testing need is very different than those two diseases. But really, the fact that they were both RNA viruses, we were able to leverage a lot of the extraction protocols we had developed to bring a test to market quicker for SARS-CoV-2.
As we deal with the impact of COVID-19 over the coming months and possibly longer, how will the company remain engaged in relief efforts?
Absolutely. So I think, you know, we're really at a pivotal point in the pandemic at this time. We're starting to see businesses start to open up, more people are interacting, and so really the need for testing is going to continue to grow. And so really we are engaging with officials locally here in Kansas City, both at a state level with Missouri, as well as with Kansas, and with other states and municipalities throughout the U.S. We really have a broad client base and serve patients throughout the United States. And so it's important. We've also recently started doing antibody testing. And so really understanding the need for PCR to, again, diagnose that you have the virus. But antibody testing, as we continue to learn more about antibodies and their role in the disease, to be able to offer that support to communities and patients as well. So in order to detect the virus, we actually measured the genomic material of that virus. And so it is a respiratory disease. So initially, to start the test, you of course need to collect a sample from the patient. And so that's most notably now done with a swabbing technique to swab either the nose, the nasal pharyngeal cavity, or the back of the throat. Really, there are different areas you can swab. And so that swab is then mixed into a transport media that is shipped from the patient, that site of collection, to the lab. And then once it gets into the laboratory, we go through initially an extraction process where we extract all the nucleic acid from that transport media. And so that's then, if the patient has COVID-19, that extracted material is going to detect, going to contain that nucleic acid from the virus. And then we go through a process of reverse transcriptase PCR, where we then go on to detect that. And so the reverse transcriptase is needed because it's an RNA virus, and so we initially do that step. And then it's really a manner of doing the PCR technique to detect the virus. You know, one of the other interesting facts about RNA viruses specifically is that they have a higher rate of mutation than DNA viruses. So most notably, influenza is another RNA virus that everybody is familiar with. And so the reason why we need to get flu shots every year is, one, is because the immune response isn't super great, kind of like we're predicting with COVID, but also there's a lot of mutations that happen in influenza. And so in order to have the best assay to detect that, one of the strategies that we use here at ViralCore is we design what we call dual target PCR assays. So we're actually measuring the virus at two different places. It's in its genetic makeup. And so that if it would mutate in one particular spot, we would have that second assay to pick it up. And so it kind of gives you a built-in fail-safe for detecting the virus.
Are you currently, who has access to the test? Is it frontline workers? Is it physicians? Or I'm sure there's a huge demand. But
sure, absolutely. And so at this point, really, the access is pretty great for those kind of that need them. We really, the healthcare system in general is kind of prioritizing testing. Of course, we always need to test sick people first. And so that always kind of remains kind of the front line. But it's very important as we get into this next phase to continue to do more community sampling to understand what the prevalence of the active virus is. And that's going to be important as we continue to plan for kind of the status of life as we know it for the next 6 months or a year. And so, of course, our vulnerable populations are always a concern as well. Those living in nursing homes, in assisted living, where they're just much closer together. So always making sure we have, we're able to support those institutions as well. If they have an employee or a resident show up positive, then we have to do much more screening at those institutions.
I'm Dina Ladd, the Executive Director for Missouri Cures, and I am thrilled to have with me today Dr. Philip Payne, who is the Director for the Institute for Informatics, Washington University School of Medicine. He is also a Professor of Medicine in the Division of General Medical Sciences, School of Medicine, and a Professor of Computer Science and Engineering at the School of Engineering and Applied Sciences. I thought it would be great if you could just give a brief overview of the Institute for Informatics. I'm honored that I'm on your External Advisory Council, and I know all the great work that you do in the Institute, but just to let everyone else know a little bit about the Institute.
Yeah, so the Institute is relatively young. We launched the Institute 4 years ago, and it was really in response to this need to grow informatics capabilities across the School of Medicine and working with our partners at both BJC Healthcare and throughout the university. And the Institute has sort of three main focus areas. One is in research and innovation, and that involves a lot of the research and development that's led by our faculty. We've actually recruited 15 new faculty in the last 3 years who work at this intersection of computation and biomedicine. Second big focus for us is in workforce development and education, and in particular, how do we train both people who will be future researchers and innovators in the field, but also practitioners such as physicians and technology leaders throughout the health system. And then the third part of what we do is really focus more on providing the data infrastructure for the School of Medicine and our partners that makes a lot of the groundbreaking research that takes place at Washington University possible, really sort of connecting the dots between biomolecular, clinical, and population health level data.
So tell me, explain how the Institute and your role are working, has such an important role in the fight against COVID-19 on the national level and also on the local level.
A number of weeks ago, we realized that as we were sort of expanding our activities to get ready for the likely surge in COVID patients, that we were really going to need this all hands on deck approach to data analytics and data science to help us make the best use of our clinical care facilities, our care providers, our resources, and to engage beyond the care system to hopefully prevent more infections than were absolutely necessary given the community spread that we were seeing. And so that led to the formation of what we call the COVID-19 Analytics Working Group, which is actually a joint venture between BJC Healthcare through their Center for Clinical Excellence, which is really the hub for quality improvement, data analytics, and reporting for the BJC system, and then the Institute for Informatics, largely as a result of the unique expertise that we had in data sharing, integration, artificial intelligence, and really more complex data visualization that help decision makers understand really, really complex and fast-moving data. And so that group has been working together now for close to a month. It's a very intensive pace. It's a 7-day-a-week, nearly 24-hour-a-day enterprise, but we have been building everything from the operational dashboards that our decision makers are using to make sense of supply chain and staffing issues, all the way through to the development of predictive models that will allow us to anticipate which of our patients are likely to become most critically ill and make sure that they get to the right care providers quickly when they interact with the healthcare system. And one of the things that we know is that getting beyond this initial patient surge and focusing on sort of what comes next will require us to be able to engage in a level of public health practice that we have not seen in this region or frankly any part of the country in recent history. And so we've been working with the county and city public health departments as well as with St. Charles and Jefferson County as well to help build data management tools to support public health workers in the field to do things like case management and exposure tracing. We've been exploring the use of mobile technologies to augment that, to allow people to volunteer their data to help us better track the outbreaks that may occur of COVID and allow us to then intervene in a very precise way. And then we're also looking at how we can combine data across all of those public health agencies as well as our major healthcare provider organizations so we can create this situational awareness of all the things that are going on so our elected officials and other key decision makers can truly understand the implications of the decisions we have to make next around, when do we relax social distancing? When do we reopen businesses? When do schools go back into session? And we really precedented things. And you may want to highlight this as part of this is that here in the metropolitan St. Louis area, the 4 major healthcare delivery systems are currently sharing data to enable the production of disease models that reflect all of our experiences and all of our clinical capacity combined. And that means these groups that are traditionally fierce competitors are basically opening up the door to look at each other's data. These organizations that traditionally compete with each other every day for patients and business basically said, we will ignore those competitive pressures and share a lot of potentially privileged information around clinical capacity and personnel and equipment and everything else to make sure that we can respond in a comprehensive way for the region.
With this issue, COVID-19, I know that you're also involved on the national level. So can you talk a little bit about that?
Yeah, that's a great question. So at the national level, one of the things that we're very deeply engaged in is a project that involves not only Washington University, but also partners at the Regenstrief Institute in Indianapolis and at the University of Washington in Seattle, as well as the National Institutes of Health. And the goal there is to actually build a large-scale synthetic data set of COVID clinical data that will allow people to conduct analyses, whether that be predictive modeling or benchmarking epidemiologic models of COVID incidents, but do so at a scale that can't be done locally. One of the challenges, despite sort of the rapid sort of increase in the number of COVID cases, is that none of us really have enough data to ask and answer all the important questions that we have. Simultaneously, sharing large amounts of clinical data at a national level is challenging because we also need to protect the privacy and confidentiality of the patients from whom that data is derived. And so we have a unique set of technologies that we've been involved in the co-development of alongside a set of partners, including a startup company from Israel called MD Clone that was brought to the US by our local partners at Global STL, part of BioSTL. And we have been working with them and the National Institutes of Health and the partners I described to stand up this national resource that will be a large-scale COVID data repository accessible to academics, individuals in industry, and citizen scientists that will protect the privacy and confidentiality of patients, but make that data available in minutes or hours as opposed to weeks or months, which is really the current situation. And so that's a very large project that we're just leading off right now. And then if you sort of look more broadly at an international level, because we do have a robust network of sites throughout the world that we work with on workforce development and other roles, we are working in close partnership with our colleagues in both China and India to learn from the experiences in China in terms of how they took a data-centric approach to managing the COVID epidemic there. And for our colleagues in India, where the epidemic is just starting to unfortunately gain some momentum, we are trying to share all of our lessons learned and work with them through a not-for-profit group that's formed of major technology CEOs, healthcare provider organizations, et cetera, to be able to share all of the knowledge that we've generated so that they can replicate those successes sometimes in some of the most densely populated cities in the world, which means all of the challenges that we faced are magnified manyfold. And I think this is sort of consistent with our belief system that from a research and workforce development standpoint, the challenges that we face with an epidemic like this effectively ignore state or national boundaries. And so we need to take a global approach if we're going to be able to get back to sort of a normal mode of operation, a normal mode of interaction. And therefore, these partnerships are really more important than ever to us to make sure that we can share our lessons learned and learn from our global partners.
As people make the decision, as governors and mayors make the decision to reopen and to lift the stay-at-home ban, do you feel that it's essential? They have to have this data to really make those decisions.
Yes. So the real challenge in front of us as we move out of this current acute phase with our various sort of stay-at-home and shelter-in-place orders, and we move into a mode of trying to incrementally reopen the economy, reopen businesses, reintroduce sort of regular interaction in the office or otherwise. In all those scenarios, the critical question is going to be, can we identify the signals that may indicate an increase in COVID transmission in the community, whereby we can intervene quickly and contain those hotspots, which is the way I would describe them, before they grow out of control. Because the sort of thin dividing line between success in reopening the economy and a second surge in patients that in some of our models could actually be larger than the current surge in patients really relies upon that level of data-driven intervention at the population level. There's a real commitment to building very quickly the data infrastructure and bringing the staff on board that are necessary to engage in that type of public health work, including creating this situational awareness of all that data and those signals so that we can make smart decisions. And part of that is, how do we make sure we get that data to decision makers in a format, at a time, and in a manner that allows them to make those decisions, right? It's insufficient to have just a large amount of data. We need to visualize it and deliver it in a way that renders it actionable. And that's a big part of what we're doing, working with all of our partners throughout the region. And I think that will be ultimately what will make us successful. And, you know, while I may be a transplant to the state of Missouri, if there was ever a time for us to live up to our sort of label as the show me state, I think this is the time, which is to actually use that data and show how that data will allow us to make smarter decisions for our population that both have economic and health impacts for them. And that's the challenge in front of us. I've said to many people, this is probably a once in a lifetime situation for many people in my field, to take what we do in an academic sense and apply it in this very real applied manner that will have direct impact on the patients and populations that we serve, and can be used by our elected officials to make the best possible decisions for the region and the state. So it is both a significant challenge, but also a huge opportunity to have real benefit as a result of all of the research and development that we traditionally do in our laboratories or in our academic institutions.
The coronavirus comes about, we are prepared. We have the opportunity to show up, and then we strike. Because who else is going to solve this problem?
That's hard to imagine, though. I think we're messing something up here.
I was an inventor as a kid. It was in my blood. Most of my success at the National Laboratory as a chemical physicist was in invention.
Yep. See, this looks fine.
When I was in high school, I really liked doing research. I made a lot of inventions.
Is it a regular cube, as we've seen before?
About 2 months ago, we started to working on this coronavirus sensor system, and we got a lot of other experts to help us.
I know it's fine, but let's make sure.
The mission of the Lightweight Technology Lab is to invent and develop novel optical and microwave sensors to further advance new research frontiers.
To invent and develop and deploy ultra high sensitive sensors. We are the de facto experts, one of maybe 10 groups in the world with this capability.
We are proposing the cornerstones of a broad project to address biohazards in public spaces, such as airport. The proposed corona sensor system actually involves 3 steps in series. The first sensor is designed to detect normal aerosols or virus-containing aerosols. After the first sensor, the aerosols will go into the second process, we call it chemical tagger. And then all the aerosols will be sent to another biohazard spectrometer.
And if we can bring this device to the airports and have people breathe into it, we might be able to say something about their state of health. You know, do they have an illness or do they not? We're taking advantage of this cylindrical cage that we walk through at many airports. Just stand there still for 3 seconds, and there's a device that scans across you. First sensor, the one that we developed, we have it coming in from the top, it comes down through a pipe and then bends over, and you're face to face with it. So while you're doing this test, it asks you to exhale into the port. And that directs your breath into the analyzer. We can take that information, feed it into the information that we get, separate out the particle size information, and just get out constitution. What is it made out of? First sensor gives us sort of a pass, no pass, where it does a pretty good job of figuring out who's sick and who's not sick, to the point where it errs on the side of, if it looks like you're a little bit sick, we're gonna flag you. And those who it deems not sick are truly not sick, like 100%. We're thinking about 30% are gonna fail. With that group, in a very short time, we used the same breath that we collected to determine that they did not pass, and gets prepared for more advanced sensor detection. If we can then use that more advanced sensor, a form of spectroscopy, to ascertain what illness they might have, then we can determine whether it's communicable, or like just a common cold, or whether it's something more serious. And it doesn't just have to be coronavirus, it could be SARS, it could be something else. We have this squiggle from the breath. It's too much to understand and to analyze. So what do? We put it into machine learning algorithms. These algorithms take all the data, and the algorithms are trained. You do a lot of breath tests. People are sick, people are not sick, etc. All this information goes into this program, and it looks for patterns.
In the near term, roughly a year, we would like to deploy the front-end sensor for clinical trials. In the long term, roughly around 2 to 4 years, we would like to deploy a complete sensor system that detects specific attributes of COVID-19 disease.
It's a tube with a rod in the center of the tube, and they can make it in a machine shop, $50. You can make it out of pieces of pipe from the hardware store. We have the analyzer for our detector. That, I'm guessing we can do maybe $10,000. It's that super spectroscopic detector that's downstream, that's charged with, is it rhinovirus, is it coronavirus? That's $125,000. The whole thing, probably around $200K. It's been vetted, people have their say about it, now it needs to be tried. Things are shutting down. How are you going to meet that need? Well, you're going to detect, you're going to confirm that things are safe.
The interconnect actually is sticking out a little bit.
You think we pushed it?
Yeah, they're not.
Okay, let's